Well, you'll be glad to know that this is the last key area in Unit 1, and this is looking at genomics and genomic sequencing. So, first of all, with this word here, genomics, genomics is a study of genomes, and in order to study genomics, you must first determine the entire DNA sequence of the organism. So, what genomics is involving is determining the sequence of the nucleotide base molecules all along the organism's DNA, and then relating this genetic information about genes to the function. Now, progress in this area has been speeded up by this here, bioinformatics. Now, bioinformatics has made genomics one of the major scientific advances in recent years. It's the name given to the fusion or the mix or the putting together of molecular biology, statistical analysis and computer technology. And that allowed us to rapidly map out and compare DNA sequences. And that has enabled us to be able to investigate evolutionary theories or evolutionary biology, inheritance, and personalised medicine. So it was a huge jump forward when computers started to get into genetics, and it meant that we were able to put together huge amounts of information and be able to map out the human genome. So the Human Genome Project began in about 1985, and it was there to determine, or its goal was to determine, the complete nucleotide base sequence of the human genome. Now there are about, as you'll see here, 3,000 million nucleotides or nucleotide bases in the human genome spread over all the chromosomes. So the project involved sequencing each of these nucleotides. There's a timeline that I'll put actually on, it was a link on here, but it won't work. I'll just add that to your iTunes U course. It's called Unlocking the Code, and I'll put it on the iTunes U course for you to have a look at. But in order to get the nucleotide sequences, then what happened was they had to cut up the sequence into more manageable sections. And that is the first step that was involved. The Human Genome Project was completed in 2003, and it has led to benefits in an understanding of genetics and diseases and so on in patients. So our first step, as I said a moment ago, was that they had to cut the genome up into more manageable sections. And they did that by using these here. They're called restriction endonucleases, or sometimes they're called restriction enzymes. And these are enzymes that recognize short sequences of DNA nucleotides, about 4 to 8 base pairs. And these are called restriction sites. And what these enzymes do is they recognize these specific short sequences and they cut the DNA. And here's an example here of a restriction enzyme. It's all called ECOR1. And it recognizes this sequence here. You don't need to know this, but so if it sees a sequence on the DNA that has got this, it will cut the DNA at that point. And there's also a restriction enzyme here called BAMH1, and it recognises a different sequence. So these restriction enzymes are used, and what happens is the enzymes will cut the DNA at every point where the sequences appear. So here we've got like 2 pieces of DNA here. It's the same piece, but what happens is that so here's, I suppose you could think of this as genome copy 1, and this is genome copy 2. It's the same genome. But what happens here is that a restriction enzyme is used here, and it cuts the DNA in different places. So you'll see the cuts that are here, and here's a DNA fragment called P. But then in this case down here, on the other copy of the genome, a different restriction enzyme is used. So you get different fragments And here you'll see, here's a DNA fragment here called Q. Up here we've got a DNA fragment called R. Now, once these fragments had been created, then what had to be done next was to actually sequence the order of the bases along these fragments. So what was here, A, T, G, C, or whatever. And that was done using a method here. It's called the Sanger method. Now, I have not posted, or I'm not going to talk about the Sanger method in this video. I'm going to post a separate video on that, describing to you the Sanger method, and it's also in your textbook as well. So watch the separate video. It's not a key area, or it's not a mandatory key content by the SQA, but it's useful to know just in case it appears in an examination. So once they had worked out what the order was of these bases along each of these fragments, what was then required was the next step, which was you had these sequences of pieces of DNA. The pieces had to be put together, and they called this way of doing things the shotgun approach, because I suppose you were blasting the genome and then you were going to put it back together. And this was done using computer analysis, looking at sections of the genome that overlapped. So, for example, here are our DNA fragments that we cut up with our restriction enzymes, and by now it's actually known what the bases are all along here, and if there's A, T, G, C, whatever. And the computer starts to look for overlapping regions on there. So in this case, the computer would recognise that the sequence of nucleotide bases at the end of fragment P matches the sequence of nucleotide bases at the start of fragment Q. And that the sequence of bases at the end of Q matches the sequence at the start of R. And it would therefore deduce that the fragments and the order of their bases run in the sequence P, Q, and R. And therefore they'd be able to know, and they would know as well what the order was of all the bases in between here. So the computer analysis of all areas of the overlap between the DNA fragments in a sample is then able to compile up a whole complete genome based on these overlaps. And obviously it would not have been possible to do this without a computer, and computer technology had been developing leaps and bounds during this time, and this enabled scientists to determine the sequence of bases for individual genes and then in turn for the entire genome. Now, it was a milestone in human history when, in 2003, the whole of the Human Genome project was completed, but in addition to actually determining the human genome, scientists also worked on a number of other organisms and were able to sequence their genome. So examples of those were disease-causing organisms, maybe viruses or bacteria, and some of these would be pathogenic, and so it was useful to know their genome. They also had a look at pest species such as mosquitoes that act as a vector for diseases such as malaria, and also there's the unicellular organism Plasmodium that causes the malaria. So, and they really looking at these organisms, we call these, they also had a look at model organisms, they had a look as well as the disease causing and the pest species. They had a look at these here called model organisms. Now these are so called because they've got genes that are equivalent to humans' genes, and they can be easily studied in the lab. And because they have got similar genes that are equivalent to humans, then inherited diseases and disorders can be looked at, and it helps us to understand any malfunctioning of these genes. So I've put on here some examples of the type of model organisms that could be used. So we've got E. coli here, looking at genetics and biotechnology, Saccharomyces cerevisiae yeast, in other words, which has got many genes actually in common with humans. And I'm not going to read out all the other ones. You can pause this and just have a read over them yourself. But these are model organisms that are used because they've got genes that are similar, have got some genes that are similar to humans. And I'm going to so just to talk about one in particular here is this cheery chap that is over here, this pufferfish. And the genome of a pufferfish has been sequenced. Now, the genome has got high density of introns and it's got very repetitive DNA. And it's an important model as it's got lots of protein coding regions and gene sequences that are common in the human genome, that are common with the human genome. And because it's got these fewer introns, it's quite a compact genome, so it makes it easier to locate the genes. So what can we do with this information when we've got it? Well, we've talked about the model organisms. We can do comparative genetics. So that's where you're comparing the genomes of members of a different species. So, for example, disease-causing microorganisms, to investigate whether they've got important genetic sequences in common. Or you might investigate members of the same species. For example, you've got the harmless strain of E. coli with the strain that causes food poisoning, to discover if there are genetic sequences or to discover which genetic sequences cause the illnesses. And it can also be used to study cancerous cells and normal cells from the same individual to try and discover the specific driver mutations that cause a healthy cell to divide uncontrollably and form a tumour. So, identifying these similarities or differences in the genomes, these could give us the clues to causes of diseases. So this study of comparing these genomes has actually uncovered the fact that between different organisms, DNA sequences of important genes are very similar from one organism to the next. And these are known as conserved genes. And examples of these are genes that code for proteins involved in aerobic respiration or for key enzymes. So it means that the close comparison of genomes has revealed important similarities. This high level of conservation, meaning that same or very similar DNA sequences are present in genomes. And much of the genome is found to be highly conserved in a wide variety of organisms and across species boundaries. And it tends to be that these highly conserved genetic sequences are those ones that code for sites of essential enzymes and things, as I said here, that are essential, like aerobic respiration. And I think I've put just a wee diagram here that's just looking at comparison between different organisms, different sections of the genome. So we're not looking at the whole genome, but we're looking at different bits of it. And you'll see it on this diagram here in this comparison. Up here, for example, here in this section here, we're looking at similarities between a human, a chimpanzee, a horse, a dog, a monkey, a rat, a cow and a mouse. And the areas that are red are the areas that are the most conserved. And you'll see that there's vast areas, like for example this section here, where it's just exactly the same. Or there's other areas that are different here, but then the other part is a conserved area. So you get these similarities. Now, highly conserved DNA sequences can be used in comparison of genomes of two groups to find out how close or how distant their relationship is. The greater the number, so the greater the number of DNA conserved sequences that the genomes have in common, the more closely related the two groups are that possess them. And you can go and have a look in your textbook at page 100 and 101, looking at shared genes and comparing humans and chimps. But there are differences, obviously, in genomes, and in most non-coding areas of our DNA, there are variations called this here SNPs. Now, that's not a campaign for independent Scotland. That stands for a single nucleotide polymorphism. And that is where you've got one base pair being substituted for another. And it's thought that there's 1 to 10 million of these, and scientists are working to locate and map them as they could help us to understand diseases and treatments for diseases. These SNPs differ from one individual, for example. So the DNA of two people, for example, might, does differ from each other because of SNPs. And I've put here just a wee thing here looking at most common types of variation that happen. This is just looking at a SNP site here where in the DNA sequence TAGC, a SNP occurs when the G base changes to a C and the sequence becomes TACC. Now these tiny variations that are there, the use of bioinformatics has enabled scientists to catalogue lots of these SNPs or SNPs and specify their locations in the human genome. And they think that this map will help them identify and understand the workings of genes associated with disease. So they're regarded as a very valuable tool in research and may aid the treatment for genetic disorders. So I've got here, it helps to answer large questions, or it may help to answer large questions involved in diseases or in cancers. Now, scientists see SNPs as a tool to improve diseases, diagnosis and treatment and planning, and we're going to just go on and talk about this. So they suspect that these SNPs may play a role in the different responses to treatment seen among cancer patients, and they think that they may also be involved in the different levels of individual cancer risk observed. And that brings us on to having a think about just how unique different people are and the fact that they explain our disease susceptibility, but they also, as well, and this is the interesting area that they're looking at, is that people respond differently to different drug treatments. And so if we could understand people's genetic or genome, then we can be looking at how we can personalise medicine more. And I'm just going to go on and talk about this here, this idea here of personal genomics. So a way back 1985, 2003, well, it took 13 years and cost millions of pounds to sequence the human genome. But it's now possible to sequence a human genome in a couple of days for about £700. And this price actually maybe has even dropped. And that opens up possibilities in medicine. So once you could be looking at predictive medicine. So you could be looking at people's DNA sequence and thinking about, well, what disease-causing mutations are there, or what mutations that are there that increase your likelihood of developing a condition. So, for example, the BRCA1 and 2 genes that are responsible for breast cancer, and I think it's Angela Jolly, I think her name is, or something like that. Anyway, I'm not really kind of all that into film stars, but she discovered that she had DNA sequence testing done looking for this gene. It was discovered that she had it, and so her chances of developing breast cancer later in life were very, very high. And so she opted to have a mastectomy because of that, a double mastectomy. And it's been well documented in the press over that. Another thing looking to the future is this pharmacogenetics, where you're looking at, well, how effective is one drug on one person and how is it affected by their DNA. So if you know somebody's genome sequence, then you could predict what medicines to use and which doses, which drugs would be more effective in one person compared to another. So there's all this field that is opening up for scientists to start to think about maybe individualizing medicine more based on an individual's DNA sequence. And in years to come, I think it will be a case that, people's entire genome will be sequenced and stored in some electronic medical record for consultation with doctors. But that's all in the future. There are difficulties in this. It's hard to distinguish between neutral and harmful mutations. So it's hard to work out whether, if you've got an altered genetic sequence there, is it really causing a harm? And diseases are obviously pretty complex as well. And some of them aren't even fully understood yet. There are, of course, a huge number of ethical issues all around advances in this field. If personal genomic sequencing becomes routine, then that predictive medical procedure, it means that you could be looking at somebody's genome that's got specific markers, genetic markers, that shows that they've got high risk of some sort of debilitating or a fatal disease later in life. And how will that affect somebody's possibility to get insurance? And that would affect their ability to get a mortgage as well. Also, as well, it might be, so I've put down here, banks and others might decline services if you've got some sort of undesirable trait, or this predictive medicine shows that you're likely to develop Alzheimer's later on in life. Or what about a company if it knows what your genetics are or your genome is, and they know that you're going to end up with some sort of the high probability or a high risk of having some kind of debilitation later on in life? Are they going to risk employing that person? That person's offspring as well? Maybe this would tell them more than they would want to know about their own genome or whatever. So there are all sorts of ethics that are involved within there. If you've got a genetic disease, do you tell your children about that genetic disease and they then will go on and get tested as well? Or is it better to keep quiet about it? There's all these moral dilemmas and ethics involved in all of this. And lots of people believe that, there should be some sort of laws and the legalization looking at introducing something that prevents genetic discrimination based on information obtained from an individual's genome. So these issues are all things that need to be addressed in the future, and maybe actually some of them just now, because there are certain conditions that can be predicted that an individual is at high risk of them, and should there be some sort of legislation in place for them.